Fantastic. Can you guys hear online?
Yeah, we can hear now. Thank you.
Oh, fantastic. Thank you for reminding me that. Yeah. And I did a whole long introduction how Rav is my best friend and we played together at school. And then, but so you're going to miss the introduction. Just to say thank you very much for joining online. Yeah. I think that we have quite a lot of participants online also. And just to say that what Ravi will do here is to talk a lot about his research on SARS-CoV-2 from the variant and immunology perspectives. Yeah. So thank you, Ravi. And let me just make sure that which screen you are seeing online. Yeah. Because today the IT managed to screw a little bit for us, but we are trying our best. I think that we have a okay display for both. So thank you, Ravi. It's up to you. Thank you.
Thank you very much. Thank you, Tulio, for that really kind introduction. And as he says, we go way back to the days of HIV, when drug resistance was an issue. I mean, I think it still is, but it's not on the radar. But certainly, we honed a lot of our skills and techniques and paradigms during the HIV evolutionary kind of exploration that we did. Of course, for HIV, we had to wait years for things to happen. And now with SARS, we see things in months or even weeks. And so I think that this is what is so exciting about being in SARS, even though everyone tells you that it's all over. And but the kind of the geeks really are now only just beginning to get interested in what this virus can do. Again, because it's really evolutionary biology in real time. So really pleased to be able to share some of our work with you. But again, to kind of reiterate that, you know, we didn't intend to work on SARS. We kind of stayed out as long as possible because we wanted to keep going in our HIV work. But eventually we kind of realized we had to do something, and that actually there were no real experts in this virus. It wasn't really a respiratory virus in its behavior in many ways. And that's why I think we were able to make some leaps in understanding. So as a sort of overview, I'm just going to give you a bit of background. Many of you will know the things I'm saying, but I guess we can discuss the complexities as well in depth. Can you everyone see the slide? Bill? Online? Online they can. Great. As long as they can see it online. Okay. So we'll do some background on virus biology, virus-cell interaction, mainly looking at the way the virus enters the cell. We're not going to spend too much time on kind of how the virus is manipulating the cell, because that's a whole field in itself. And then I want to sort of speak about evolution, but also the phenotypic aspects of it, as well as the genotypic. Okay. And then to sort of focus on some of the key biological characteristics that we've kind of that have become important. Again, mainly looking at entry phenotypes, and then of course what this means for vaccines. So there's another nice thing about SARS is it brought together the fields of virology and immunology and genomics, you know, very, very closely, and people who had not worked together much before were suddenly, you know, looking at different sides of the same coin. And with this virus, you couldn't really look at one without the other. So I think that was also really interesting. So here's some sort of diagrams of the virus. It's an RNA virus, of course, 30 kb genome. It has a glycoprotein on its cell surface called spike, which everyone knows about. And if you look more closely at the interaction between spike protein and the ACE2 receptor, which is found in many different cell types, and this is one of the problems with SARS and why it has a very wide tropism for cells and why it causes multi-system disease, is because its primary receptor ACE2 is found in so many different places. The glycoprotein has these S1 and S2 fragments, and importantly, it's not often discussed, but the S1 and S2 is cleaved in the producer cell, should be cleaved by cellular furin, a bit like HIV-1 GP160 is cleaved whilst it's being before it gets assembled on the cell surface for export and for new variants to come out. So there's a cleavage event that happens between S1 and S2, and that's called the polybasic cleavage site. And this enables the S1 and S2 to rearrange. You can see now they've been broken up they reassemble in a different configuration, which is then competent for binding ACE2, and then the subsequent fusion step at the surface membrane. Here you can see once the ACE2 has been engaged by S1, the receptor binding domain, you get some conformational changes that reveals the fusion peptide in S2, and that inserts into the target cell membrane. When that happens, you bring the virus membrane close to the cell membrane, allowing the fusion event to happen, and then the virus RNA to enter the cell. Here's a sort of these are cryo-EM representations of what the spike actually looks like. It's of course asymmetrically distributed on the virion surface. And the other important thing to kind of understand is that the spike exists as trimers. In other words, there are 3 identical polyproteins that assemble together. They have intermolecular contacts that need stabilization and lead to overall stability. But the interesting thing is that those trimers can adopt different configurations. So there's this thing called open and closed conformations. This is absolutely critical for spike function, because if the receptor binding domain is closed, in other words, it's kind of brought inside, it cannot engage ACE2 and it can't enter a cell that way. You can have different combinations of the number of trimers that are up or down. So you can have all 3 up, or 2 up and 1 down, etc. And the reason that there is a dynamic flux is, of course, because of molecular dynamics, but there are reasons for the virus to keep the RBD down, and that is, of course, because of immune evasion. So if the RBDs are exposed, that is, of course, the immunodominant target for our antibodies. So the virus is playing a balancing act between keeping the RBD open or up for attachment, whilst trying to evade antibodies, and therefore it doesn't really want to be up all the time. So that's why we have this situation. And I think that evolution is driving spike to maybe change the proportion of the time that it's in the open or closed conformation. Looking more closely at that receptor, that interface up there in the top right, you can see just some of the contacts between ACE2 and spike, and you'll recognize these as also areas where some of the key immune escape mutations are occurring. So that's again another important thing to recognize, which is that the very interface where the virus needs to make contact for cell entry is also the area where most of our, or many of our antibodies are generated against. So again, the virus is playing a balancing act between engaging the cell receptor and avoiding these antibodies. So some of these mutations that you'll have seen, the N501Y and E484 changes, or K417, that are seen in many of the VOX, are at this interface. And so so on occasion, you can find that the immune escape mutation will have a detrimental effect on the ACE2 binding. So this is just mapping on the different domains. I've already mentioned the receptor binding domain, which is the one everyone talks about, really, which is up here in the open conformation with an antibody kind of bound to it. You've got an N-terminal domain, which is actually displays some of the greatest variation across different variants. So it's really really plastic, and there are a lot. It's one of the most mutated sites in the virus genome. And in fact, in other coronaviruses, the N-terminal domain is the actual part of the virus that engages the receptor. So the NTD in some viruses, in some coronaviruses, is the thing that's actually doing the engaging. So in terms of evolution, it's a really important area. We don't really understand that well what it's doing in SARS-CoV-2. I mentioned the How's that? Can everyone still hear me? Yes? At home? Or in the other offices? Wherever you are.
Yes.
Thanks. So yeah, we've got the NTD and RBD. These are the S1 components up there on the top left. And then you've got the fusion peptide in S2. And the cleavage site between S1 and S2 is kind of in the middle there, which my professor is not showing there. That's fine. But anyway, between the orange and the green. And that's where the cleavage event happens. And then, of course, you've got the S2 region over there on the far right. So No, it's okay. I think maybe just. So let's move just on to a bit of mutation. I know this, being in this audience, you all know this, but very briefly, in the early days, everyone was quite obsessed with this substitution rate, and it was measured around two substitutions per month, which was fairly modest, I think, and in keeping with a virus that was had a short incubation period, rapidly transmitted, a lot of asymptomatic infection, and so this sort of hit and run kind of approach to infection and transmission was consistent with a low selective pressure, you might say. Also, there's a proofreading mechanism in the polymerase, so the chances of getting mutations are relatively slim theoretically. Because of proofreading, we know that HIV-1, for example, or HIV or lentiviruses in general, don't have proofreading mechanisms to enable them to generate more mutations. So all of this contributes to theoretically low selection pressure. If you put that in a framework of what we understood about antigenic variation or in viruses, we had this drift scenario. Again, influenza is the kind of archetypal example of this. On the left, you can see how viruses drift. This is a segmented genome, of course, that influenza has, and you just accumulate mutations over time. The way that influenza generates large changes in its diversity is through shifting. We call it antigenic shifting, and the way that influenza achieves this is through manipulating its segmented genome. So when two viruses that are different infect the same cell, they can reassort, they can mix their segments. In other words, to produce a progeny virus down at the bottom that has a mixture of genome segments from A and B, and that's how you get these different combinations of H's and N's, hemagglutinin neuraminidase. So that's how sort of flu has caused pandemics, through this reassortment. Now, if you think about SARS-CoV-2, it doesn't have a segmented genome, so it theoretically shouldn't be able to shift. This is a virus that should have been drifting at this substitution rate of 2 substitutions per month. In other words, we should have been seeing phylogenies like this, and this is from a review paper published just this year that shows this sort of the sort of drift scenario with these viruses with short branches here, and the genetic distance from the root, of course, follows a straight line in general. So there's over time, so there's sort of gradual accumulation of mutations. But of course, what actually happened was more like this, where we had these long branches appearing apparently from nowhere, leading to new virus and new sort of antigenic variants that then followed the drift pattern afterwards. And so if you plotted those, you get this kind of picture where there's sort of lineage A here, and then the new variant having a similar accumulation rate, but of course it's shifted from here to here because of this long branch in terms of genetic distance. So so that's what we were observing, which was kind of weird. If you plot this in a what we call an antigenic map, so these have been developed from the influenza field again, and what these show is different viruses and how well they are neutralized by serum from an animal or a human infected with a certain strain. So the bigger the distance between a circle or a box and another circle or a box, it shows you the antigenic distance. This is the loss of ability to neutralize that virus through antibodies mainly. So this is this antigenic cartography, as we call it, is a measure of how well antibodies against one virus can neutralize or block a virus that's more distantly related. So you can see, if you just very quickly here, these are some of the early B lineage viruses, but you can see the Omicrons out here. So there's quite a big distance between the original ones and these Omicron viruses. And even with an Omicron now, you can see very big differences or distances between them, consistent with the fact that we're getting all these different waves of infection. In other words, all the subsequent waves of Omicron are escaping immunity, and this is the demonstration of that fact. So it appears that SARS-CoV-2 can appear to shift antigenically, but then the question was how, because of course it doesn't have a segmented genome, it can't just swap big chunks between viruses. So how is it achieving these big jumps in mutations or groups of mutations? And I did have a slide here that shows the 3 possibilities, which are one, that it's just very fast accumulation and you're not sampling the patient, the individual, so you never see all of the stepwise mutations, or that there's an animal reservoir and there's back and forth between animals and humans, or the third possibility is chronic infection. I think personally, and most scientists believe, that chronic infection with SARS-CoV-2 is the explanation for how variants of concern arise. And we were one of the first groups to propose this because we we observed in our hospital a case of chronic infection in mid-2020. And this individual was immune compromised. The individual had a lymphoma previously, had been treated with chemotherapy in the past, was currently taking rituximab, which is a monoclonal antibody that blocks B cells, and in other words, renders you with very, very, very low levels of antibody. And so this individual had a chronic infection over 100 days, which is 3 months, something like that. And there are even longer cases described now. But interestingly, in the first 30 odd days, in the first month, this graph plots the abundance of particular mutations by NGS. Here is the sort of variant frequency. In other words, this is a sample of nose and throat swab with a particular mutation at 100%, all the way down to 0%. So we sampled around 23 different time points in that individual, and we measured and we performed whole genome sequencing, and then obviously measured the abundance of different variants within that individual. And you can see nothing really happened in the beginning. Then the patient received a remdesivir, but more importantly here, two bags of convalescent plasma with polyclonal antibodies. These were, of course, plasma samples that were taken from recovered individuals who had had COVID-19. And when those were infused into our patient, because we didn't have any treatments really at that time apart from remdesivir, when that was infused, we suddenly, after about a lag of something like a few days, we saw the emergence of mutations, and this is just showing the spike mutations. Two of the mutations were the D796H mutation in the S2 fragment, and then a deletion in the N-terminal domain called the 6970 deletion. And these hadn't really been described before. Yeah. Who did the sequencing? We did the sequencing with, I think it was actually Department of Pathology. Yeah, at that time. Yeah. Yes, we did it basically in-house. I can't remember the reasons now. It's all a bit of a blur. But Sanger weren't involved in this in this stage. But we used the Arctic protocol for it. Yeah. So importantly, the 6970 turned out to be a really important mutation. We'll talk about it later because it was what was called the S gene target failure mutation. There was a diagnostic test that was used for measuring viral load, and there were three targets, and one of the targets was around the 6970. And viruses that had this mutation were noted to only have two positive signals, and the third one had dropped out. And the reason it dropped out is because the viruses had deleted that region. This was very early on. And then what was interesting is that those mutations faded away over time. And then when we gave further dose of plasma, containing from a different donor, but probably containing some similar types of antibodies, we saw a re-emergence of that initial population of viruses. So the selective pressure had selected a virus population. The antibodies faded away because when you infuse antibodies, they don't have the half-life is around 2 weeks. So when they disappear, another population comes up, and then when you reapply that pressure, you reselect those mutants. So we thought this was very good evidence for selection by antibodies for SARS variants within an individual. That was just spike. But looking at whole genome level now, this is what was happening between days 82 and 101. These are all different mutations in different colors, at different abundances, frequencies in the patient. The reason I'm showing this is just to show you the sheer number of mutations that were happening outside spike, but also that groups of mutations were going up and down. So this is evidence for haplotypes. In other words, populations of viruses with a whole range of mutations in each virus population, and they're going up and down in groups because this is virus A and this is virus B, for example, and there's a virus C somewhere out there, whatever. So there's an intense competition going on within the body. We're really sampling the nose and throat, of course. So if this is what's happening in the nose and throat, you can imagine what might be happening in the gut, for example, where we did find virus, or in other organs. So just showing you what's going on, and we think much of this potential was triggered by the antibody treatment, but we can't prove that. So we then had the opportunity, because we're a molecular biology lab, we then tried to start figuring out what these things are doing, what these mutations were for. And this diagram shows you the N-terminal domain up there, which is where the deletion was. It's out in a loop. It's actually not resolved in cryo-EM structures, and it can't be predicted. So it's kind of in a disordered loop in the NTD. And then the S2 mutation here, 796, we see this mutation in the Omicron variant as well, and in a number of other viruses. It's again in an exposed loop, and it may have a role in antibody evasion. But we wanted to figure out, whether that was true or not. So we used technology from the HIV days, where we generate pseudotype particles. We use lentiviruses that are HIV on the inside. In other words, they have a capsid, they have a reverse transcriptase, but they are coated with spike glycoprotein. So we can measure entry phenotypes, and we can measure sensitivity to neutralizing antibodies with these particles, and you can do it in BSL-2 rather than 3. So these are the Western blots showing that the viruses have HIV P24 inside them, and that they're expressing spike. And we made different mutations in the spike protein to represent what we saw in the patient. So you got a single mutation at 796, the N-terminal mutation here, and then the double mutation at the end. And then we measure infectivity on target cells. And what you can see in the corner there, hopefully, is that the deletion, 6970, had around a 2-fold enhancing effect on infection, consistent with this being an enhancer mutation for fitness. But the 796 actually was quite detrimental. It was sort of almost a 10-fold drop in infectivity. So that is a bit curious. Why is a mutation making a virus less infectious or impairing its entry? But I guess the answer then is, of course, you're looking for immune escape. Maybe that's an immune escape mutation. And so what we did next is we took these convalescent plasma samples from the bags that we gave to the patient. We took the serum from there, and we tried to measure how well the antibodies in the serum or the plasma were able to neutralize these different mutants. So the key thing here to see is that the 796 has a fold change above 1. In other words, in each of these cases, it's about a 2- or 3-fold loss of neutralization sensitivity with that mutation. The deletion I showed you that was an enhancer has very little effect. It's around 1, all of them. So the enhancer does not affect neutralization sensitivity, but the other mutation did make the virus relatively resistant to the antibodies.
So both together is more and body infection.
Yeah, that's right. So that's right. So really, what probably happened is that this mutation arose first because it was trying to escape the antibodies. Then the virus was not happy because it was a bit defective. So then it selected this mutation deletion to enhance its infectivity. And interestingly, we saw this in I don't know if you remember the virus that came from mink. This virus had Y453F. It's an RBD mutation, and very soon after that escape mutation emerged, that virus acquired the deletion 6970. Another virus, the Scottish variant, had another RBD mutation, also then acquired 6970. So I think we'd basically explained why that was happening. So that was a classic paradigm for SARS, right? Immune evasion because the host is generating antibodies either through vaccination, previous infection, or exogenous administration of antibody, and then the virus has to compensate for the immune escape mutation, causing a fitness defect in its life cycle. Further evidence came from here in South Africa. This is work that Tula was involved in, as well as colleagues at ARI, and this is in the context of HIV. Where an individual was followed for 6 months and acquired this range of mutations over time. Again, change at 796 that we had seen, but also other mutations that are seen in Omicron. Many of these are Omicron mutations. And when they made these antigenic maps that I was talking about earlier, this is another way of representing such an antigenic map. See that the early virus over there in purple became a late virus over there, and it had shifted from there to there in terms of escape from the serum antibodies that were generated early on. Those antibodies early on were not able to neutralize the late virus. So this was kind of, again, further evidence that chronic infection can generate variants of concern, really. And so I don't think, in my mind, there's much of a debate as to where these variants are coming from. And there have been, since this, many other case reports. Okay, so chronic infections are the likely source of variants. Immune-compromised individuals appear to be the main source, because all the reports I've seen have been in people on chemo or with immune deficiency or with HIV. And that's a problem because the pool of these individuals is huge worldwide, and it's also very difficult to do something about it. So with that, I'm just going to go through a few key topics. One is immune evasion, one is replication, and also one is about tropism shifting. And these are properties that, of course, variants of concern have acquired. And I want to give a slightly historical perspective, because I think with all the talk about new Omicron variants, we kind of lose sight of how this all happened. And I think that it's also the sort of evolution started in a more a simpler way, you might say, so with fewer groups of mutations occurring. And what we're seeing now with Omicron is far more complex in many ways. So, I mean, if we go back to the early days, of course, again, the alpha variant that emerged in the United Kingdom and the beta variant that was first detected here in South Africa, and then, of course, the gamma variant that was first detected in Brazil, had this common mutation in the receptor binding domain at position 501. And of course, two of those variants had further changes at 484 and 417. And as I showed you earlier, these are the key contacts between the ACE2 protein and spike. At this stage, people thought, okay, we now know how the virus does its immune evasion. Of course, subsequently, we realized it's more complex, but certainly this is how it all started. And the important thing, of course, to notice is that there were multiple other mutations in different regions, and we didn't really know what many of those were doing. We were very much focused on what the receptor binding domain was doing. As a good example of that, the alpha variant had the deletion 6970 that I mentioned earlier in its internal domain. But the other two main variants there did not have that deletion. So we set out to, I mean, at the time it caused a lot of worry, if you remember, whether the alpha variant was going to lead to a failure of vaccines. And so we used our pseudotyping assay for SARS to show really that the loss of neutralization against the alpha variant was actually very small. So each of these is an individual who's donated blood and given serum after their vaccine, and we take those sera and we mix that with the viruses that I showed you, and then we test how well the antibodies have blocked that virus. And so the higher the number, the higher the neutralization for effective the antibodies. We're looking for a change to go down. If this went down, it would suggest that the virus is escaping. Really, most of those lines are kind of horizontal with a small drop. So suggesting that when you compare the Wuhan virus with the alpha variant, there was very little change in neutralization. That was good news. Plotted over here in another study, this is a alpha variant B117 compared to wild type, very small difference. But then, of course, when they measured the beta variant here, there was a much greater drop in neutralization sensitivity, and that caused obviously big worries worldwide. And that was corroborated by the trial data, some of which came from South Africa, that showed that vaccine, the ChAdOx1 vaccine, was only 10% effective at stopping transmission with the beta variant. So that was pretty worrying for most people. Of course, the vaccines were then shown to confer quite good protection against severe infection, which was the main endpoint. We changed our endpoints as time went on. If you remember, we all expected vaccines to give sterilizing immunity. Yeah. Yeah.
Alpha variant, to be honest, the 501 mutation was the one that.
Yes.
Mm. 501. That was the mutation that allowed the.
Right. Yeah.
That's why we give them 501.
Yes.
Straight away, they the alpha, and then they accused South Africa that we had the variant. Don't understanding that vaccines.
Yeah. Yeah.
But the alpha went to spread like
Yeah. Yes, that's right. Alpha caused a lot more morbidity. That's for sure. And then I guess what was interesting is people kind of thought we knew the answer. All these variants are going to have N501 changes, but then of course Delta came, and this virus didn't have N501 changes. It had different RBD mutations. It had this L452R mutation, and then the T478K in the RBD. So totally new paradigm in a way. So this is what kept us all kind of engaged and interested. We were thinking this virus has different routes to immune escape. And of course, with only 2 mutations in the RBD, it caused obviously devastation worldwide. And one of the key and one of the big questions is why. So of course, it also has a bunch of NTD mutations, which I may talk about earlier if I have time to talk in those slides. But you know, we didn't know what any of those were doing. It also had a really key change in the cleavage site that I talked about, the S1, S2 cleavage site. It had an enhancer mutation, and of course that polybasic cleavage site is the area where it was touted as being the evidence that this was an engineered virus by various people. But of course, the polybasic cleavage site is found in many coronaviruses, and Delta had basically changed one of the amino acids at position 681. That meant it had enhanced cleavage. So that may have contributed to its increased infectiousness. So, but when we did the neutralization work in the middle, you can see that the wild type is in blue, and then Delta is over there in green. There is a drop. In other words, the virus is more resistant than both the alpha and the wild type to antibodies generated from vaccination. So this virus had managed to generate immune escape with only 2 mutations in the RBD. But probably a significant contribution from the NTD as well. We were fortunate enough to be working with colleagues in India at the time, and they had done a study in 3 different hospitals. They had vaccinated healthcare workers with ChAdOx1, the AstraZeneca vaccine, and then they had taken blood samples, and also then they had been sequencing or collecting sequences from breakthrough infections after the vaccine in their hospitals. And so we worked with them to analyze their data, and we found that during that period, there was a whole range of different viruses circulating. And these B viruses, you can see B117 in brown, and then the 617.2, the Delta variant. And what we noticed is that the big clusters were being generated by the Delta variant, suggesting that the Delta variant had extra qualities. It was either more transmissible or it was immune evasive. So that correlated very nicely with the increased escape from neutralizing antibodies, number one. But we thought there was something else, which I'll come on to later. We showed in vitro that the virus was actually more infectious. It was about 100-fold more infectious than alpha in various systems of cell culture. So the Delta variant had acquired two new sets of properties over and above alpha: more transmissibility, more infectiousness, and secondly, more escape. Then, of course, fast forwarding to Omicron, this was a major step, you might say, of course. Yeah, in terms of mutational load, you can see the Omicron there with the red flags just show you the different mutations or the numbers of mutations. You can see huge number of mutations compared to Delta above that. And mapping that onto the structures here, you have Delta here and Omicron there. So mutations in all the key regions, the NTD, which is in green, the RBD, which is in purple, and then the S2 region, which is in red. So huge numbers of mutations in different domains. And at that time, of course, everyone was again worried about what the vaccines were going to do. And by then we had boosters, we had dose 3 going around. What was very interesting and clear is that that after 2 doses, the neutralization against Omicron was really poor. Second dose, neutralizing titers were only around 100, which is for Delta it was around 1,000, and for wild type it was probably 10,000. So we went from 10,000 to 1,000 to 100 in terms of neutralization against Omicron. So really immune evasive. But after dose 3, something interesting was happening. So 1 month after your dose 3 with an mRNA vaccine, we got really quite nice titers, around 10,000 against Delta as you'd expect. But against Omicron, we reached titers of 1,000. And that's using a vaccine that was really quite distant from the actual infection. And this was suggesting that the immune system was somehow broadening its coverage in terms of the target, the spike protein. And there was a nice paper in Nature sort of explaining how this was happening. And it's due to potentially due to something called affinity maturation, which is something that happens in germinal centers in lymph nodes after you're vaccinated or after you experience an infection, where the immune system continues to select for a breadth of antibody responses over time, and particularly after you receive booster doses. So the booster doses are able to potentially drive that process forward. We don't really know if this happens beyond dose 3, and I would be skeptical to sort of say to believe that it would continue in a sort of stepwise fashion. But certainly after 3 doses, you were getting a broadening of responses that may explain why the data I showed you earlier. And that's lucky for us because, of course, the Wuhan vaccine provided very good protection against severe disease against Omicron. So the challenges around vaccination, of course, are obvious to all of us. One is age, and as we get older, our vaccine responses to everything will diminish. You have waning responses over time. Of course, responses do drop, and the titers drop, the frequency of circulating cells drops as well. And then, of course, the problem of immune suppression and how do you generate good responses in people who are on chemotherapy, for example. We contributed to some of these areas. First of all, in age, I mean, mainly because we have a big elderly population in the UK, and we were able to show that if you're over the age of 80 and you received 1 dose of an RNA vaccine, the probability of response, in other words, generating a neutralizing antibody response, really drops off at the age of 80 really, really sharply. So up until then, you have a gradual decline with age, as you might expect, but there seems to be this kind of very big drop-off after that age. Fortunately, after the 2nd dose, some of that is restored, but there is still a significant difference between those less than 80 and those over 80 for different variants. So the 2nd dose is really important in the elderly, we found. And that correlated again with protection because we still saw cases of very severe disease after dose 1 in the elderly individuals, certainly in Europe. And so 2 doses was critical for them. Just moving away from neutralizing antibodies for a moment, I think I will touch on T cells in a minute, maybe. Of course, there isn't very much evidence for T cell escape, and I think that doesn't mean that T cells are not important. It just means that T cells may be providing a function that either is dampening overactive immune responses, or that they're targeting highly conserved regions that don't mutate. Just wanted to touch a little bit on innate immunity because it's often forgotten, and we've got interested because we wrote a review recently. Most coronaviruses have, of course, they've got such large genomes. They have multiple proteins that are designed to overcome many different arms of the innate immune system. This is just a diagram showing you some of the pathways involved. So coronaviruses are known to activate things like the CGAS sting pathway. They activate MABs and a variety of other sensors, but also viruses encode a number of different proteins that can overcome them. For example, ORF3A, ORF7, furthermore NS1. So multiple parts of the interferon and the innate immune pathway are activated by infection, but of course through evolutionary selection, there have been significant gains of anti-immune function in the viruses. So one important sort of set of discoveries is around the fact that SARS is also evolving to overcome innate immunity as well as antibody and T cell and antibody immunity. So this is a paper just showing you that ORF6, which is one of the ORF9 and ORF6 are known interferon antagonists, as in the previous slide. And you can see here that the number of transcripts, the number of RNA reads in the virus has actually enhanced. So the viruses are making more RNA templates of these ORFs that are known to overcome innate immunity, suggesting that this is for alpha, showing that alpha here in pink is upregulating key interferon antagonists like ORF9b and here So that's interesting. And the mechanism behind that is interesting. It's the viruses are changing sequences in the lead of the TRS sequence, which is just upstream of the coding sequence. So in other words, allowing more templates to be generated. I think so. This second bit is where we're most interested in. I mean, replication is kind of where most of the virology is, but it all starts with ACE2 binding. And of course, we all know from the media that one thing that SARS has been doing is it's been busy optimizing its affinity for the ACE2 receptor whilst trying to evade antibody immunity. So these plots over there on the far side show you the binding affinity, and what you're looking at is how steep the decay is, because and that represents the off rate. So once the virus binds, that's called the on rate, and that's usually fairly quick. But what determines binding strength or affinity is more how slowly they dissociate from each other. So you can see Omicron has a very slow decay here, and the other ones are much steeper. So basically, Omicron shows that Omicron has evolved to hold on more tightly to ACE2. And that's using a method called biolayer interferometry, which is a fairly standard biochemical assay. I mentioned earlier that Delta was much more replication competent than Alpha, and these are some of the data that our lab produced show that. So you can see here that in lung cells, that the Delta variant over time is generating much higher levels of virus compared to the Alpha variant. Log scales. You can see at 20 or 48 hours, it's about 3 logs. It's 1,000 times more virus in culture compared to Alpha. You can do similar experiments in air-liquid interface models. So this is mimicking the nose. Put nasal mucosal cells in a well. So there's air on the top and liquid on the bottom, just like in your airway. You can put the virus on the top layer, representing the airway and infection, and then you can measure replication in the liquid part. Because it's porous. And so when you do that over time, you can also see the Delta variant in green, it far outcompetes the Alpha variant in terms of the amount of virus being produced. And finally, on the right, we've got a system called where we use organoids. So these are essentially derived from stem cells, and then they associate themselves into actual mini organs. So those are alveolar cells or lower airway cells on the right. And they have holes in the middle, they're tubes. Containing all the relevant cell types, and you can actually infect those systems and measure virus replication. And again, you can see the green bar is much higher than the orange bar, suggesting that Delta has outcompeted or is more effective in that compared to Alpha. So we have different model systems now for measuring how different variants are going to compete with each other on infectivity scales as well as immune evasion scales. And these are really important sort of things to have in the lab to be very responsive and quick when new variants emerge. Infectivity is not all about the spike protein. Of course, I've been obsessing on spike, but there are lots of scientists out there working on different parts of the virus. Nucleocapsid has been shown to also increase infectivity. Again, we think it's by incorporating more copies of virus RNA into the particles. From this nice paper, it was one in Science and one in Cell Host Microbe from groups around the world, where they showed that the mutations at position 203 and 204 in nucleocapsid have been recurrently mutated in any of the VOX to enhance the ability of the nucleocapsid to grab onto RNA and take it into a new virus particle. As many virus particles won't have RNA in them just through random processes, but if you increase the affinity of nucleocapsid to RNA, you're more likely to obviously take the virus genetic material into the virus particle. Last bit is about tropism shifts. This is kind of, I think, the most interesting, as well as fusogenicity. So I'll spend the last few minutes on that. SARS-1 and 2 have been known to have 2 different entry pathways into a cell. One is through, and I really only showed you the first one, which is surface entry fusion over there on the right. That's what HIV is thought to do as well, where the virus basically engages the receptor on the cell surface and then releases the nucleic acid material into the cell. There is another pathway also open to other viruses, which involves endosomal entry. So you get engagement of ACE2, but then the virus is swallowed up inside the cell. There's acidification of that endosome as it goes inside the cell. That acid environment triggers cathepsins, which are proteases that are sitting inside endosomes, and those things chop up spike protein to mature it and to allow fusion. So what is the relevance of that? Well, the relevance is that, first of all, we all noted that Omicron was less severe than Delta. The second question was why? If you looked at in animal models, these are data from Syrian hamsters from my colleagues in Japan, where they infected the hamsters with Delta or Omicron. So the Delta is in orange, and it's the severity index, different severity measures, but in every case, the Delta is higher than the Omicron in green. So in animal models, in the lungs, Delta is also much more severe than Omicron, mirroring human data. And we showed in our lab that if you try and compare replication of Omicron and Delta in the nasal system that I showed you up here, that they're actually pretty similar in kinetics and titers. So in the nose, they're pretty much the same. But if you then look in lung cells or organoids that represent lung cells, there's a big difference. The Omicron is really impaired compared to Delta by, again, 1,000 times. So there's some kind of defect that Omicron has when it's trying to infect lung cells, and that probably explains why we didn't see so much hypoxia or severe lung disease with Omicron. We can test that further using drugs that block one of those pathways or the other pathway, and we believe these results show that block the plasma membrane fusion route, then you are able to block Delta, in other words, inhibit. This is percentage inhibition. You can reach 100% inhibition of Delta fairly easily just by blocking cell entry route at the top, plasma membrane. Omicron is not so effective. You can only inhibit half of the 50% of the virus. You get the opposite result if you try and block the endosomal route, the other pathway, suggesting that Omicron likes to go inside the cell and Delta likes to just fuse at the surface. And the relevance of that finding is that the plasma membrane route is more commonly found in lung cells. So if I take you here, these cells are dominated by the plasma membrane route, and that's why Delta is more effective there and Omicron 4. And in the nasal cells, it's pretty much 50-50. So in lower airway cells, the plasma membrane route is required. We don't know why, and therefore a virus which is inefficient at entering that way is not so fit. Okay, so just wanted to then sort of summarize that to say that Omicron was quite radical. It's a radical event, not just because of multiple mutations, and not just because of immune escape, but because it had shifted tropism from lower airway to upper airway. So it's kind of adapting to become a proper respiratory pathogen, right? The classical respiratory pathogen, rhinovirus or all of this common cold viruses are upper tract infections. Yeah, you don't get pneumonia usually. So SARS came to us as an accidental transfer. It's now adapting because it figured out through evolution that the most effective way to transmit is to just infect the upper airway and to transmit that way. But I think that the shift from the lung to the upper airway is some kind of evolutionary mistake, because there isn't really a selective advantage on the virus, we don't think, because the patient dies usually 3 weeks after infection, let's say. By then they've transmitted to everyone they were going to transmit to. So the death, the mortality is not related to the transmission, we don't believe. So we think that this has happened as a side effect of the mutations it's acquiring to escape immunity. So this is a problem, of course, because for sterilizing immunity, because you need, as the virus gets better at transmitting in the upper airway, it's harder to stop it. And so our aims at mucosal immunity or generating nasal vaccines has become much harder, because this virus is now even more highly adapted to transmission in the upper airway than before. The other thing I wanted to touch on that's related to tropism in our minds is syncytia formation. This again is a property of many viruses. So most viruses with an envelope to a glycoprotein, even HIV, can form syncytia. And in fact, this is showing you HIV infection of macrophages. These are macrophages without infection. When you infect the macrophages, they start fusing together. So what's happening is that the HIV glycoprotein then traffics to the cell surface, and it engages the CD4 receptor on another cell, and then the engagement causes a fusion of membranes. Very similar to when a virus will infect a cell and cause a fusion of membranes. So similar process happening, mediated by the surface glycoprotein of the virus. And of course, it was realized that SARS can do this. This is a SARS-induced syncytium. The reason I'm showing you all this is because in the early days when people were dying and they were having autopsies, people were seeing these big giant cells, in their lungs. They were seeing fused cells. And we think this is a marker of pathogenesis. So we think that this ability to fuse cells is related to how bad or severe the disease is. And we think the reason that this happens is because the human body has evolved systems to detect cell-cell fusion. Because viruses do this, the body has now, or humans or mammals have now figured out how to detect it as a danger signal. And what happens is you get triggering of interferon-stimulated genes when cells detect cell-cell fusion. And that, of course, can lead to severity because it leads to inflammation and lung damage. What was important is that the Omicron variant here developed an assay to compare Omicron to Wuhan, for example, and Delta. And if you measure over time the rate at which virus cells are fusing together, you can see that Delta fuses really, really well compared to the wild type even. But Omicron is unable to fuse cells together. And that potentially also relates to its milder clinical disease profile. I won't say much about Omicron and its further evolution because it's now getting very complicated. Just to say that what was interesting in Omicron is that the first successful recombinant, I think, was the XBB. It was truly, truly successful. So I think recombination may be something we see in the future in terms of evolution. And then just to summarize, we think that variants that are antigenically shifted have arisen from chronic infections. This is kind of a new paradigm, I think, for a respiratory virus. So it's something we've never seen before. That's a tick. These variants can compromise vaccines, and we've been very lucky that vaccines protect against severe disease, and that's probably due to T-cell mediated immunity, I would have thought. So although T-cells don't get much airtime, they're probably the reason that our vaccines are still working. Omicron has done something really radical. It's shifted its biology through multiple mutations. I think one of the kind of golden sort of targets or aims for us as a lab would be to figure out what's the mutational trajectory, or what mutations arose and why did they arise in order to get to the Omicron variant. And we may get that information from studying chronic infections in individuals where we can see the stepwise accumulation of mutations. And then we can phenotype those viruses at every stage of that process. Okay, just in future, I think we want to know what the limits for spike evolution are in terms of immune evasion. We keep seeing new mutations, even though we think we've reached the summit. We're interested in allostery, which is the effect of one part of the protein on another. So spike is 1,200 amino acids long. We've got data and papers showing that the N-terminal domain affects things that are happening in other parts of the virus, and that's through allosteric molecular dynamic changes. And I think that's important because it's the big barrier to genopheno prediction. So there are people out there saying they can predict what the virus is going to do from just the genotype, but actually you can only predict some things and not most things, actually. And so we really need to understand allostery if we're going to make our models for prediction better. Of course, vaccination has huge barriers at the moment. How many doses do you need? Which variants are going to be in your new vaccine? And then, of course, can mucosal vaccines actually be the solution? These are the guys that did the work you've been seeing. So without them, of course, this couldn't have been possible. And of course, thanks to our network of collaborators around the world. And I'll take any questions. Thank you. I've overrun a bit. I'll blame that on the long introduction.
Yes. You can blame on me, no problem. Yeah, fantastic lecture. One of the big characteristics of our seminars is after the presenter is to grill the presenter and see if what he's presenting really grow true. So I think that we try to generate a lot of discussion. That's where a lot of the interesting part come. So maybe let's open for questions here in person, and then in the people online. Yeah, we can already start playing. But as you think here, I already have Richard Lessels that want to grill Ravi Gupta. And please thinking about the difficult questions here for Ravi. So Richard, try to unlock your or unmute and go on.
Richard, before you start, just remember I could tell the
I'm just glad Ravi came back after our latest incident on the tennis court. But Ravi, to what extent do you still see the chronic infections now back at home in the UK? Is it still something that you observe, or do the kind of antivirals and things work effectively to clear the virus in these patients now?
Yeah, that's a really important question. Thanks, Richard. We are seeing chronic infections, certainly from our own data set, which is an immune-compromised population. They haven't been so prolonged, so we've been seeing, infections out to sort of 30 to 60 days. But then, as you say, many of those people are immune suppressed, and they will get some kind of treatment, like a monoclonal antibody or something like that. So I think some of our treatments are stopping us seeing some of these chronic infections, but I believe they're still going on, which is why we're getting new variants. Yeah, but I think surveillance is as important as ever for these chronic infections to understand what the next level of trajectory is going to be for the virus. Thanks, Richard.
Yeah, just to continue from Richard. So you think that the new Omicron BA2.86, that's the one that hit the news a month ago with this large evolutionary step. Do you think that's from a chronic infection? How would you be able to prove that?
Yeah, that's interesting, isn't it? I mean, as Tulio knows, that virus has roots in the in BA2, right? And it seems to have, if you phylogenetically, it seems to have come directly from BA2. So it's been sitting around somewhere in someone or some animal or human for a while and accumulating mutations. So I think that one definitely has good evidence for a chronic infection or some kind of the word is maybe occult kind of evolutionary process somewhere where there's no surveillance or in an individual that's not sampled, of course. Yeah.
Thank you so much for the talk. You mentioned some skepticism for the effectiveness of the fourth booster. Can you comment on, the design of future vaccines? Like, should we be targeting both means of entry, or how would that affect? Would immune evasion then be a bigger problem because of that, or would we solve the SARS transmission problem?
I'm not sure we're going to solve the transmission problem, well, unless mucosal vaccines work really well. The problem with them, I think, is how long you can keep those responses up in the nose and throat. So you can vaccinate and get really great levels, and then they probably just drop away. And the other studies have shown that you can get really good levels in the airways, but then the lymph, the levels in the blood are not so good. And maybe it's the blood levels that give you the longer term, or the lymph or the tissue levels that give you the longer term protection. So I think that there's some work to be done there in terms of mucosal versus injection, and maybe you do both or something. I don't know. Yeah, but the vaccine field, I think, again, this virus has opened up some really amazing and interesting and challenging questions in the area of immunity, right? And again, people might think it's not that relevant now because the death rate from SARS is so low, but maybe the next virus that comes will be much worse. And we'll be sitting here with, vaccines not being good enough and people still not surviving, and then these questions will be pertinent, right? That we really do need to stop transmission now.
Yeah, thank you, Ravi. I was wondering, the graph you showed for this case with chronic infection with these, where you give plasma and then one variant comes up, which comes back a few weeks, I think. I don't know what the time frame was after that. So I was wondering, with HIV, we know it integrates and there's a reservoir. So how does that work in SARS-CoV-2?
Yeah.
Is it just prolonged infection of lung cells, or where is this virus then coming from a few weeks after that?
This is a great question. Love it. In fact, that's partly what this is talking about. This is all about reservoirs. I didn't really use that word, and it's a word that we throw around in HIV a lot. But when you talk about an infection over 6 months, there's a reservoir there, you know, and this also comes back to when I said that the actual theoretical mutation rate is very low for this virus. The fact that we see so many mutations being selected tells you that the number, the effective population size, must be massive, right? For this virus, it must be enormous, so that even a low mutation rate can generate all of these different things, you know. So yeah, there definitely is a reservoir. Again, the tropism is related to this. The fact that you have ACE2 in your liver, in your gallbladder, in your gut, in your brain, this represents virus in different compartments. And of course, immune surveillance doesn't happen so well in all of those compartments. So you have the opportunity of virus to evolve independently. In fact, we have a paper that we are writing at the moment, where in this patient, we sampled nose, throat, and then trachea at the same time, and about 3 time points. And we found quite big differences in virus population just in that distance there on the same day. You know, different mutations, for example, up here and down here. So yeah, there's definitely a reservoir, and whether it's a source of variants, I think it's contributing to VOC generation, yeah.
And maybe a more broad question. So for SARS-CoV-2, we have benefited a lot from HIV field, but HIV field has been like very quiet in 2 years. So how is HIV now benefiting from all of this work done with COVID?
Yes, a great question. I mean, I went to the AIDS conference in Brisbane this year, and I was amazed at how much work had been done. I mean, obviously, this is like a 4-year gap where you didn't see anything, but there was stuff going on. I mean, still being presented and really good advances happening. And I guess technologically, there are advances that are feeding back. I mean, you know, use of single cell RNA-seq, things like that. The immunology is feeding back. I think that there should be the knowledge about, spike conformational dynamics has to feed back into HIV-ONV, I think, if people can do that. And again, so I think, yes, there's a lot that will be learned from it for HIV. It might take a little bit of time to realize it, and we'll have to look back and say, oh, we figured that out because of SARS. So I think that it will happen, as long as enough people move back to the HIV field.
Okay, let's get more questions here. People that.
A microphone. But I think
thank you so much for the presentation, Prof. We have also tried to do a bit of work on immunosuppressed patients and these chronic infections. And one of the things that is happening now is sort of in our South African context, people are really no longer just bothering about testing anybody. And I was wondering, are you still seeing such cases in the UK, and what can you advise us within the South African context, especially knowing that we've got such a huge burden of HIV and people that are likely to be immunosuppressed?
Yeah, I think that one of the things, I mean, obviously optimizing HIV care, I think that's what SARS has kind of reminded us of, is the importance of sustained viral suppression in HIV. Obviously, diagnosis, number 1. Number 2, getting ART initiated, and then 3 is maintenance of suppression, and that's where drug resistance also kind of feeds back. And it's quite sobering to think that lapses in HIV care over the last decade could have been responsible for generation of variants. I mean, it's a sobering thought of the consequences, the global consequences of what you might call local differences in care provision, investment, all of these things, education, that could have a global impact. So I think, without the stigmatizing, if you take the stigmatization aside, it really it just brings home, of course, the globalized nature of everything that happens, and small effects in one place can have big effects globally, that are long lasting. So in terms of what can we do, I think is your question, right? Well, I mean, yeah, you know, HIV care is kind of hopefully coming into the era of long-acting therapies, but I mean, all of these technologies rely on diagnosis, right? And reducing transmission. So I think a renewed global commitment to ending HIV/AIDS, I think, is needed, because, as you say, the pool of immune-suppressed people due to this disease is enormous, and it's reversible and it's preventable. I mean, we may not be able to prevent cancers, but we can certainly prevent and cure HIVs, and limit HIV progression to AIDS. Yeah.
Okay. Yeah. So yeah, more questions?
Sorry. Yeah. So these chronic infections, particularly respiratory infections in immunocompromised patients, we know for other viruses as well. I think.
Yes.
Are you already looking into other respiratory viruses? Are we not recognizing maybe the role that immunocompromised patients have in evolution in other viruses?
We're not, again, we're really slow at changing fields, to be honest, and we kind of get stuck in one area. I mean, the true future seers are already doing, you know, as you say, you want to do metagenomics on immune-compromised people. They're looking at things, and so I think that that is one of the next frontiers of understanding, and using, again, the knowledge from SARS to kind of guide that investigation. There are lots of things we see clinically, and people die of diseases in the hospital. They're immune suppressed. We think we know the answer as to why they died. We often get it wrong, and often we don't have the cause, especially in respiratory infections. It's very hard to get samples from the lungs. And so, I think there's a lot to learn, certainly from doing sequencing and characterization of those virus populations. And the technology is there now. I mean, you know, so to do multiplexing and metagenomics, so it's great. Very exciting area.
Yeah, we had a big crew online. Yeah, with people online. Yeah, any question? Yeah, sorry. Any questions for the people online, please? Okay, so just one more or two questions. Sorry. When we bring them here, so they know that it's not just for the free lunch. Yeah. So one question: your immune suppressed individuals. Yeah. What was the cause in the UK? Yeah. And the second question: why on the beta, on the minus, on the people with more than 80 years old? If you show the after the vaccines seem to work even better than the alpha. Is that not puzzling for you?
So first was about immune suppressed people in the UK.
The ones that you analyze. What
we have. We're doing a study at the moment on vasculitis. So people treated with rituximab, this B-cell depletion agent, and really remarkably, I mean, we're finding that people don't respond until dose 3 or 4 of vaccine. In other words, you can't neutralize virus until dose 4. And these are young people in the UK who are taking this drug. What we do show, which is interesting, is that they generate antibodies. They have a lot of anti-SARS specific antibodies, but they're just not neutralizing. So again, we're learning something about the immunology behind some of that. So that's one area: vasculitis, autoimmune disease. Then, of course, we have renal transplant, diabetes. I mean, the pool of immune-compromised people is very, very significant. Many diseases now require disease modification with, you know, for example, irritable bowel disease or inflammatory bowel disease, rheumatoid arthritis. These are all treated with immune-suppressive drugs. So that, you know, it's a huge population. And above the age of 80, this is actually something that's described in the field of aging, which is that changes in biological age are not always linear. There is a drop-off. I don't think they quite understand why. And the actual age of drop-off for different parameters is a bit different, but it's around that age, somewhere between 70 and 80. Colleagues of mine have done studies using lots of biomarkers and correlating those biomarkers with outcomes and health and disease. And there is this kind of predictive drop-off that happens. If you have a certain combination of biomarkers, then you end up dropping off. Something happens in terms of your overall health. It may be related to inflammation, for example. So inflammation.
Yeah. And why the beta has quite a high. effectiveness.
Oh no, this is the
vaccine,
percentage with neutralization less than 1 in 20. So this is the percentage with no neutralization. So the percentage with no neutralization is highest for beta because it was the most immune evasive. That fits.
General. Okay.
Yeah, that fits with the known kind of escape. Yeah, it's just showing you that the age compounds that problem, because in beta, many people are not responding, like more than 50%, even in the young group, as well as the old group, the older group.
Okay, thank you. Any more questions for Ravi? Thank you, Ravi. Not only your presentation was great, but you more than tried on your grilling questions. Yeah.
Did I pass?
Yes, you pass. You can come again. Yeah. And just my last announcement. Yeah. First, thank you so much for Ravi. I think that he deserves another big round of applause. Yeah. Yeah. So from anyone that would like to have CPD points, continuous professional developments, we do offer here. There is a registrar outside. You have to register with your name and your HPCSA number. And the same for people online. Yeah, if you could put your HPCSA number there in the chat, Mambele is going to be doing, and your name, so we can register for you. But it seems that we don't want to let the presenters to go. So we have here Eric Miwangi with the hands on. Eric, would you like to ask the last question?
Yes, thank you so much for this opportunity. There is a question that I was asked in the chat about the Prof has talked about the drifting and shifting of the virus, of which is the mutation. So we understand that mutation is due to the deletion, translocation, or insertion of the genes. For the case of co-infection of SARS-CoV and HIV, we understand that both of the two RNA viruses.
Thanks.
Should I continue?
I was just going to read from your thing. It says whether HIV can, whether you can get whether HIV could insert segments of genome into SARS. Is that right?
Yes, exactly. Yes.
Yeah, during a co-infection. Yeah. So is it theoretically possible that you can get insertion of genomic segments from one virus to another? I mean, I think the answer has to be theoretically yes, because we're seeing segments of human genome integrated and inserted into SARS. But the question is, which of those is what is compatible with increased viral replication? Most of these insertions, due to copying, may end up nowhere, or they may be deleterious. If you look at the life cycle as well, if you think that if HIV, we now think that reverse transcription is really happening after in the capsid, so it's protected. So there isn't really the opportunity for the RNA in HIV to interact with SARS RNA, because SARS RNA is generally going to be in the cytoplasm and in these what are they called again? DMV, double membrane vesicles. And the HIV genome is going to be in the capsid, which then goes to the nucleus, protected in the capsid, and then it uncoats in the nucleus. So there isn't really opportunity that I can see for that to happen. If you then think about RNA export, you get exported spliced and unspliced RNA. Maybe that could encounter RNA from SARS. So there are places where things could happen, I guess. But the point is that 99.999 of those events will be deleterious. And so I guess, yes, it's not impossible.
Thank you.
Great question. I think that's a really important question to understand where these evolutionary boundaries lie. I mean, we can see that gene duplication happens in viruses. We can see that viruses have obviously evolved new proteins, over time. Have they co-opted them from other viruses? These are important questions. Yeah, I mean, the other thing is endogenous retroviruses. Other places where you can potentially swap segments from.
Thank you so much.
Okay. Okay, thank you very much. I think that's time to give a break to our presenter because we want him to come back. Thank you. Thank you so much, Ravi.
Cool. Thanks very much.
Yeah. And anyone that want to talk to Ravi, we're going to have some finger food outside, and you're very welcome to engage with him. Thank you so much for coming. Yeah.
Hey, enjoy that.
Yeah. So we.
It's good news about Sanger. Are you going to take this thing up?
Don't give Ravi food poisoning again.
Hello.
What is Dr. Lessos? Are you challenging him
Don't
on
give
a
him
tennis?
food poisoning.
Oh, food
Got
poisoning.
food poisoning the last time he was.
And no tennis.
No tennis.
I thought that you have a public challenge on the tennis on live TV.